The microbiome of animals, of mice that we would buy from a commercial vendor, essentially all academics and pharma and industry would use, all come from several or a handful of large commercial vendors that produce these animals. And what they've done over many decades is systematically deplete the microbiota of animals, largely to sell a reproducible product of an animal that has a very defined microbiota, so that people can essentially have the same research tool over many years or across different sites. You can sell to different universities and industry partners and have them reproduce everyone else's data. But in doing this, they had essentially separated or even divorced the animal from the contributions of the gut microbiota. Again, this started many years ago, before we really even appreciated how important gut bacteria were to development of the immune system, metabolic system, nervous system, and other systems. And so the idea would be is that instead of using this very depleted and uniform microbiota to model a diverse human population, perhaps we can develop animal models which are more reflective of humans by transplanting microbiota from humans into mice. So this way, we're not only introducing more complexity in terms of the species of bacteria, but also viruses and fungi and all the other things that we have that laboratory mice don't have, but also to develop a panel of animals where it's not just one microbiota that is representative of humans, but rather a panel that covers the broad range and diversity of human microbiome. So we all know that we're different in many ways, but we, as humans, have a certain space, if you will, in terms of what our microbiomes will look like. And essentially, that's the platform that we built at Nuanced, was to leverage human microbiomes in well-established mouse models to see if we can both recapitulate human phenotypes in animals, but gain more insight on what may, in terms of drugs or other treatments, that what may be more likely to work in humans because they were screened in systems that were more similar to humans.
And, our goal is to start representing clinical diversity in the systems that we use to discover and develop drugs. And by doing so, we hope to be able to uncover some of the medicines that we believe will be more suited to work for everybody. And I think one of the key theses of the company, and really the basis of what we founded the company on, is that we know that human biology is diverse, and the way that this diversity manifests itself in the human population is that we have two people that have the same disease, but that disease looks completely different. The outcomes are completely different, and similar to drug responses, where two people with the same disease take the same drug, but they respond completely differently to that drug. And when Sarkis and I sat down and thought about, what is our process currently to discover and develop drugs, is, it really became apparent that throughout the whole process, we really take a very reductionist approach to the discovery and the development of drugs. And what I mean by this is that, even when we start out in the process of discovering what target to actually drug, we take these complex and diverse human populations and we reduce that complexity, reduce that diversity by taking averages of populations. But even when we start to identify targets and identify molecules that hit those targets, we study that biology in a very reduced complexity manner, where we take these in vitro systems and really study biology in a dish. And right before we go into human clinical trials, really a bottleneck and a necessary process is to show efficacy in mouse models. But even when we go into these mouse models, every mouse model that you choose depending on what disease you're studying, you're ultimately studying a very homogeneous mouse population. Like Sarkis was saying, we systematically and strategically reduce the variability of these mouse populations, and what that means is that we don't really get the chance to understand or to even interrogate clinical diversity until it's almost too late when we're in clinical trials. And what this has produced is a process that fails 90% of the time. And even in the best of successes, where we get an approved drug, we know that response rates very rarely reach above 50% efficacy in the population. And so, our goal at Nuanced Health is really to systematically reintroduce this biological heterogeneity and diversity back into the preclinical models. And the lever that we pull on this is humanizing the mouse microbiome. And, we really believe that by doing this, we can start to address that barrier to success, which is the inherent clinical diversity, human biodiversity that exists at the very beginning of the process. And by identifying what are the drivers of clinical diversity, and understanding and leveraging these mechanisms, these underlying mechanisms of disease and drug response through our mouse models, we really believe that we can pursue novel drug opportunities and unlock maximum potential from the R&D process, meaning the efficiency and the efficacy of the R&D process and the drugs that are produced from them. And so really simply, our platform takes diverse in vivo meaning in the mouse, diverse in vivo clinical outcomes. And these diverse outcomes are in the form of disease progression or disease outcomes and drug response. We collect a very large suite of bioinformatic data, which is very high dimensional, from genomic data to molecular data, and put that all together to identify novel therapeutics, identify how to make current therapeutics even better, to identify new biology, and then ultimately hope that all this together can enhance the inefficiencies or enhance the efficiencies that we currently face in drug discovery and development.
It's always fascinating to hear the latest and just what you guys are doing. I think one thing that I always find interesting is just a little bit of that origin story. Just maybe because that's just getting the science out, getting that initial inertia. That's a huge part of this. So just maybe walk us back to that point where you decided, because you were in Sarkis's lab and you guys had been collaborating on this, and why this idea? Why did you make that leap, and how'd that come about?
Yeah, and so like Sarkis said, I had a couple opportunities to join a startup full time before I joined grad school, and my dad, being risk averse, told me that maybe I should further my education a little bit, and so I took his advice. And always from the beginning, thought about academia, thought about industry, but really thought about industry in the context of building something. And Sarkis was really, I was fortunate that he allowed me to take a summer and go to Flagship Pioneering, where I was a fellow there for a summer, and there I really got the experience really thinking out of the box on how to translate fundamental basic biology into these really unique companies, and came back from that knowing that Flagship, who is successful in their own right, is building companies that are founded by recently minted grad students. And so came back in Sarkis's lab and kind of immediately told him that I wanted to do something, and thinking about the different options that we had. Like I was saying, I was really struck by the multiple sclerosis study that I was a part part of, not only in that the microbiome is interacting with multiple sclerosis, but that the microbiome fundamentally reprograms the biology of mouse models. And that in and of itself was something that I didn't appreciate before. And given how widespread the use of mice are, and given our dependence on the understanding of biology and the understanding of drug response is on the mouse, really saw this as an opportunity to improve the entire R&D process altogether.
Gotcha. And for you, Sarkis, you had a few, I think, startups beforehand. So, why do this one? What made this one interesting enough to dedicate your time to, and maybe walk through, how your involvement has continued with the company.
Sure.
Because that's interesting for our audience to understand how the PIs, how the principal investigators are still involved.
Sure. Yeah, I think I had a few motivations for both thinking and then ultimately executing on what has become Nuanced. But it was really, as I described, this opportunity to leverage the science that we were doing. Again, we have a certain expertise and knowledge base in the laboratory. There's many things we don't know, so we want to really stick to our strengths, and that's trying to understand the functionality of the microbiome. So, folks in the audience may be familiar with the fact that you can order a kit these days and put a sample in there and send it out for DNA sequencing. So a lot of that technology going on in many labs that are both doing the sequencing and then the informatics to essentially deconvolute what the data from DNA sequencing will tell us. But and I would say that's a large part of the microbiome, both in terms of what's in academia, but also what's in industry. But our expertise is a bit different, right? So we're more interested in the function of the microbiome. What are the organisms producing? How are those molecules they're producing interacting with cells? How are they interacting with tissues? How are they mediating disease outcomes, right? And so the opportunity to leverage that in a way that both, as I described, improves the translatability of animal models, essentially makes them more similar to humans, but also really this notion of the fact that, we could hopefully, and the mission is really no less than potentially to revolutionize the way drugs are developed and drugs are discovered, right? And if we can do that in a way that is more efficient, that is more reflective of outcomes in the clinic, then I think that this is, a technology that many should adopt moving forward. And it's also the risk, right? I like to take some chances, and because I think that when you take those risks, if they're well formed, that the payoff may be larger, right? And as I said, this is a really an untapped area. We don't know of anyone else who's doing anything quite similar to Nuanced.
David, I'll jump in with a question that's sort of a segue, Sarkis, to some of your comments, and Bryan, yours as well. And the notion of revolutionizing drug discovery and development is pretty grandiose, and it's an out of the box, moonshot kind of idea. But look back to some of the data, Bryan, that you shared, and take into consideration the fact that 90% of new molecules fail at the clinical trial stage, and your data shows relative to the more standard Charles River, mice versus the Nuanced mouse model. Your data shows that it's knowable a priori which subpopulations won't respond to a certain drug, and it's also knowable which subpopulations will show the complete response. So this question is really to both or either of you. If you could unpack that a little bit more, because I actually, and David and I both subscribe, Sarkis, to your notion that this really could revolutionize things, particularly the critical step from preclinical animal studies into human studies, which currently show a 90% failure rate.
Yeah, maybe I could start by, I will caveat that the proof is ultimately going to be in the pudding, as far as how successful we are at approving drugs, which is more of a long-term goal. But what I will say is that the amount of biology that we've learned through mouse models and the amount of, you know, drug mechanisms that we've learned through these mouse models, all being incomplete, is quite a bit. And so the entire premise that we haven't actually addressed how these mouse models can be diverse in and of itself until, you know, the formation of Nuance, you know, was a little bit mind-blowing to me, and to be able to really start to unpack some more of the foundational biology of disease and the foundational biology of drug response by understanding how microbial diversity implicates all of that. You know, I do think that although, you know, human data and translation will be the ultimate measure of our success, that at least currently, you know, we will undoubtedly be able to unpack a lot more of these underlying drivers of diversity that we really had no capabilities of knowing this beforehand.
And maybe I'll augment that with a different view, a complementary view, is that oftentimes, not exclusively, but oftentimes when there's a new hypothesis, an untested idea, it's certainly, you know, maybe not feasible or not ethical to test that idea directly in humans. And so we start with simplified reductionist systems like mice. And then if you see a result and then you develop some insight into what may be causing or mediating that result, potentially towards a mechanism of action, you do it all in the sort of the mouse system, right? Without ever having any idea of whether or not those discoveries translate to people. So then you maybe marshal the resources and the collaborators, if you have to then look to get a glimpse in humans of whether or not there's similarity to what you initially discovered in mice, and this could take many years. But what I think one of the superpowers of Nuanced is that we start, as we've described, with human tissue. But we know a lot about those people as well, or we can know a lot about those people, right? Did they respond to a drug? Were they obese, right? Did they have a cancer that was resistant or sensitive to a current therapy? And then you start with that human metadata, and you take the human sample and then you put it into the animal. And now if you can recapitulate that human result, because again, essentially the project started in humans, then you're much more likely to believe that you're indeed modeling something that's relevant to people, right? Without having to cross your fingers going from mice to humans. And again, when you do this now in not one person, but many, many people, you can start understanding what are the differences. Why do certain people respond or look differently than they do? And, you know, to what degree does the microbiome recapitulate that functionality when transferred into mice? And at least it gives you, you know, the vector that is relevant to that outcome, because again, we're not doing anything else other than transplanting the microbiome to humans. So that whatever similarity we observe between humans and mice in this reverse translational scheme is dependent on the microbiome. And then we have the tools to then dissect and really understand what it is about the microbiome that's mediating all that clinical diversity.